Hello everyone, this is MHM Mubashir again. Welcome to the BTC509 Bioinformatics tutorial. In this tutorial, we'll be downloading the structure that we have predicted through Swiss model, and we will validate the structure by different servers. Okay, so to download the structure, remember we'll always download 3D structure or protein structure as PDB format. So to download this, you have to click here. After clicking here, it will download all the 4 models as a zip file, and then this is the folder that contains all the information of those particular 4 proteins, which is EXA21. So these are the images that you might need, and then these are the models. Okay, so like I said to you that we have 4 models. So the first one, you see the model, this is the PDB file, and if you have installed any PDB viewer in your PC, then you can view this. Like for this, I have installed Rasmol. This is a very easy software. So if you Google Rasmol and download it, you can see the structure. Okay, so I'm closing it. So similarly, we have some other structures, like for this one, this is the second model, and for this one, this is the third model. Even you can see the image of that model, say for this one. So these are the 4 models we have got from Swiss model. And out of 4, like as we have seen that the first model, which qualified best in the Swiss model server, will be analyzing and evaluating the structure's quality within different servers. But before moving to that, I'll be modeling the same protein using some other different servers. So let's do this. So I'll be using 4 different servers besides Swiss model, which are Geno3D, RaptorX, iTezer, and HSpread. Let's do this. So first, Geno3D. So if you type Geno3D and press enter, then it will take you to the server where we can model the EXA21. So the database, we should select NPS 3D sequences from PDB for this case, and then the name could be like EXA21 here. So we'll be pasting the sequence from this part to this part, and then we'll paste the sequence here, and then we will just submit. Okay, so it's simple as it is. So it will take some time, and then let's move on to some other servers of protein modeling. Next one I want to Google is RaptorX. Okay, so RaptorX is another server. So here you should click RaptorX structure prediction. So I'm clicking here. So after clicking it here, we should give a job name, like say for example I am typing EXA21 Raptor, say RaptorX, and then you have to provide your email address. So I'm providing mine. So say for an example I'm giving my Gmail here. So this is the sequence of EXA21 I have just pasted. Now let's press submit, and it will send you the result to that particular email address. So its status is pending. So within this period, I'll be going to some other server, which is iTezer. iTezer is a very good server for predicting the structure of a protein, but remember you have to open an account here. So say for an example, if you don't have any account or like password, so you have to click here and do register for this. So I'm clicking it here. So you have to give your first name. Say I'm providing my first name here, and then I am providing my last name here, and then the institution should be like BRAC University. So here the Gmail will be declined, so you have to use your BRAC email ID. So for my case, I'm using mhmubashir@bracu.ac.bd, and similarly mhmubashir@bracu.ac.bd. I'm using this. So here you can see that I have received an email from iTezer. So I have registered as mhmubashir@bracu.ac.bd, and they have given me a password. So I'm clicking it here. I'm just pasting my sequence here, sequence of EXA21, and I am providing my email address mhmubashir@bracu.ac.bd, and then you have to provide a password. This password has already been sent to you to your email address, so you have just copy and paste it, and then you have to run iTezer. Okay, let's look into some other servers. So for in case of Geno3D, you have to select the template. Say for an example I'm selecting the first template. for this case, and then I have to run the job again. So select template, and then I have to give my email address here in case of Geno 3D, and then I have to launch Geno 3D here by clicking this. So it says modeling job, this is running. So you'll receive an email after pressing the launch, and then after completing the prediction, this server will send you the email with the structure. Okay, so let's wait for a few minutes for all the jobs to be done. So let's move to the final server, which is HSpread, for predicting the 3D structure of our protein. So this is my personal favorite, and this server is one of the best like I have seen. So the name of the server is HSpread, and then just type HSpread and press enter, and here we have the server. You just need to paste the faster sequence of X21 here in the server, and then you just need to press submit. Well, I have given the sequence earlier, like few days back. That's why it is saying that we found an identical copy of your job in our database like 2 days ago, but I want to start the job anyway. That means I want this server to do the job again for me. So start job anyway. So it will take some time. Like Swiss model, this server will do multiple sequence alignment and will figure out the possible templates that we can use for modeling our protein X21. Then we can manually select one or more templates for this server. That means in the previous servers that we have submitted our sequence, that was for single template modeling. That means they are modeling the protein, or they are predicting the structure based on one single template. But for this server, you can submit or you can select multiple templates, right? And I'm advising you to select not more than 5 templates, because HSpread server, they use the algorithm of modeler, and it is advised by the modeler creators that you have to select from 1 to 5 templates and not more than 5 templates. Okay, so we can select 1 to 5 templates for modeling our X21 protein manually. Also, like Swiss model, you can tell HSpread server to select the template automatically based on its algorithm. So you can do it both automatically and manually. Well, so we have got the result, the initial sequence alignment result of X21 by this HSpread server. So you can see that they have selected the sequence from 77 to 630 automatically, because they think that if we want to model this X21 protein, modeling from this portion to this portion would generate the best result. Well, okay, so let's talk about these templates. Okay, the first templates, we can see it here, is definitely the best one for modeling the X21, and like so on. Okay, and obviously we can see that this template is not covering the entire sequence. Okay, this is one protein and this is another protein. Also, these two can be both of a part of a single protein, because like this area you can see is the transmembrane region, and is very difficult to model, or it's very difficult to generate the crystal structure of a transmembrane region of a protein. So that's why they couldn't model, or they couldn't generate the crystal structure of the transmembrane region for all the proteins. Okay, we can have one protein here, which is, I can see they might have modeled the transmembrane region. Okay, well, so these are the sequence alignment result that we have got. So they have generated 250 results for this case. Now, if we want to model the X21, say I want to model the X21 based on this template. Okay, and obviously we want to select the first one, because this could give the best result for our case. And say for an example, I want to select multiple templates. Say for an example, I want to select template number 1 and then template number 2, and then here, say for an example, I want to model the entire protein. So select the entire one, and let's select 6XR4. So I'm selecting 6XR4 as well for modeling the transmembrane part. So I have selected 3. I can again select 2 more, which is for kinase domain modeling. Let's select 2QKW, this one. Let's see if we have 2QKW here. So 2QKW we can see. So I have selected 4 templates. Let's select another one, the 5th one, 5LPB underscore A, 5LPB underscore A. This one. So now you just need to click here, model using selection. So whenever you'll be clicking here, then it will start generating the model, one single model for your protein HSpread. But the problem is, as I said to you already, that this HSpread server uses the algorithm of modeler. That's why you need a modeler key for this job to be done. And I'll be providing you the modeler key in the live session, so don't miss that. And as I have already saved my modeler key in this HSpread server, I don't need to give this again. Okay, so it will generate the model automatically. So they're saying that these are the information that has been generated for our proteins for modeling. Now just forward to modeler. Okay, and you see it is saying that modeler key is stored in your profile, so just you need to submit. So it will take some time for modeling the protein. Also you can see that we have submitted the sequence of HSpread in this server RaptorX yesterday. It's been 24 hours, but still our job is currently being processed. But we have got the result of Geno3D. Okay, so let's see the result of Geno3D. So Geno3D has generated 10 models, and let's see the result report here. So you can download any of the models by clicking there, but I'm not doing so. I just want to see which model could be the best for us first. So here they have model energy. The higher the negative value is, the better for us. So for this case, I think this one, the model 6. Also they have given you the Ramachandran plot summary result here, and definitely the model 1 is not good because the disallowed region is 7.2 here. So what is disallowed region, what is allowed region, or what is core region? I will be talking about this in the next video, because in the next video I'll be talking about the validations of our proteins or the predicted structure. So these are the results that we have got. And for model 6 we can see that the disallowed region we have 1.6%. So let's just download the model 6. Okay, so I am downloading the model 6. So just click it there and it will automatically start downloading your model. At the same time, we can see that HSpread has completed generating the modeling. So I can download the PDB file here by clicking it. So I'm downloading the file. Okay. So after downloading it, if you click it here, you can see the model of HSpread. You can see that, okay, it has modeled the LRR region. It has also tried to model the transmembrane region and kinase region. For the other modeling tools, like we have downloaded the structure of Geno3D, it couldn't model the entire sequence. This prediction is not correct, but at least it tried to model the entire protein, right? But for Geno3D, it couldn't model the entire protein. For RaptorX, it won't be able to model the entire protein, so far I know. Still, I'm not sure when I will get the result of RaptorX. Same goes for iTezer. We still haven't received the result of iTezer yet. It sometimes takes 2 to 3 days for the result to come. So let's wait for that. And also for Swiss model, you saw that the Swiss model also couldn't generate the entire protein sequence. That's why if you want to model a multi-domain protein, you should follow the approach of multiple template modeling. Okay. In the next video, I'll be showing you some validating tools, and also I will say in detail about the Ramachandran plot and how we can have a very clear understanding on our predicted structure, either that structure is good or not. So thank you everyone. Have a good day.